Hello and welcome to this video on homebrew clearing agents. These are also known as fining agents. Fining agents are used to clear homebrew of yeast, sugar and other contents that are not readily dissolved. These have a negative effect on the appearance, mouthfeel and flavor of the brew. A large part of fining removes protein, as was demonstrated in the earlier video on Isinglass. This video builds on the content of that video and explains some alternatives. The raw ingredients contain the largest proportion of protein and other contaminants that are removed by the use of fining agents. Other sources include the yeast selected and waste products produced during fermentation. Yeast cells break down, and the cell wall is largely composed of the bipolar protein-lipid complex. Waste from the breakdown of the substrate, and sorghum is particularly well known for this characteristic. The first option in removing haze and other signs of protein in beer, wine and cider is the use of low-protein ingredients. This is much easier to do when brewing beer and grain-derived spirits, as these have a better profile of each ingredient used and its composition. Cider and wine are less convenient, as these have less well-profiled ingredients and are often more a personally familiar source of material, whether this is an old apple tree in your yard or an orchard nearby that's friendly. The next option is the addition of charged products, such as Irish moss at the end of brewing. Other options include commercial fining agents, Isinglass, gelatin, and diatomaceous earth, although the last is more obscure. The first option is cold settling and racking. Racking is the moving of your brew from one vessel to another, leaving any sediment that is at the bottom behind. As a further step in racking, or as a means of clearing your home brew, cold is a good option. This is called lagering when used in association with beer, but the principle works with most home brewing. The cold environment causes all of the energy in the vessel to decrease. This decrease in energy causes everything that was floating in suspension to sink to the bottom, just like how oil and water separate. Or when you add salt to water and leave it to sit for quite a while, you'll find that salt builds up as crystals in the bottom of whatever container you are using. Calcium is another useful tool. It is used in beer brewing particularly, but it also works as a clearing agent which causes yeast to flocculate. Calcium does this over a broad pH range. This occurs through an increase in the amount of calcium leaving the yeast cell. This efflux leads to linking of cell walls and the yeast falling. In addition, things like citrate ions inhibit flocculation. This is done by sequestering or locking the leaked calcium away. Flocculation by salts other than calcium is restricted to neutral or high pH ranges. Therefore, the flocculation of any brewer's yeast strain is dependent upon the presence of calcium to one extent or another. The minimum expectation or requirement would be 200 micromoles of calcium chloride, and this is almost enough to ensure complete flocculation of yeast. The next option, and perhaps easiest to use, is the use of high flocculation yeast. Flocculation is defined simply as the rate at which a particular yeast strain will fall out of the beer, ethanol wash, wine or cider, and any other kind of fermentation once the fermentation is complete. If you choose a yeast strain with a high flocculation rating, it will clear much more quickly than one with a low flocculation rate. Flocculation should not be the only consideration, but if you have a choice between something that is highly efficient with flocculation and performs just as well as another product, pick a yeast strain that has both matched to your style and has a medium to high flocculation. It is a very complex process that depends on the expression of specific genes, the transcriptional activity of the genes, and is influenced by the nutritional status of the yeast cell and other factors. Flocculation is also controlled by factors that affect the cell wall composition or morphology. Yeast flocculation is governed by the competition between the electrostatic repulsion and the polysaccharide protein bonds that make up the cell wall. Electrical surface proteins, which are mainly due to the phosphodiester's linkage of the cell wall, maintain the cell's dispersal, and the polysaccharides and proteins readily penetrate these potential barriers and establish bonds between cells. Bottom-fermenting yeasts respond to the pH of any wash, and this leads to a higher water phobicity than top-fermenting yeasts do. This in turn has an effect on the flocculation of bottom-fermenting yeasts. Flocculation of bottom-fermenting strains is governed by the lectin-mediated mechanism, and it is inhibited by mannose and some other sugars. It requires calcium specifically, and occurs in a narrow pH range different from the isoelectric point, and is not influenced by the amount of ethanol in the wash. Flocculation of top strain is not inhibited by mannose, does not require the addition of calcium, and takes place at the cell's isoelectric point. Low concentrations of ethanol broaden this pH range in which the cells flocculate, and it is favoured by an increase of ionic strength. Ethanol may induce flocculation by reducing the electrostatic repulsion between cells and by decreasing the steric stabilization. This allows the protrusion of certain polymers into the liquid area around it, which then bind to the protein wall of other cells. As with Isinglass, most of the defining agents discussed so far work through electron charge, either a positive or negative charge. Seen here is a simplified protein structure. The different parts have a positive, negative, or neutral charge. Brewer's yeast appears to flocculate or disperse reversibly in response to environment's conditions, and the yeast's solubilized cell surface substances show flocculation dispersion changes according to the pH, from which it is apparent that it has a bearing on what substances should be used to clear it. The defining agents have a charge, examples of which are Bentonite, which is negatively charged, egg whites, which are positively charged, Gelatine, which is positively charged, Casein, which is positively charged, and activated charcoal or carbon, which is neutral. Use of defining agents should be considered for several reasons other than removing contaminants, and these include storage stability and appearance. The removal of protein in particular has the direct effect of removing possible amino acids, and they're a source for an infection. This reduces the viability of your wash sustaining a population of infective agents, or keeping the population low. It removes excess sugar, which denies any infection access to a foodstuff, and it has removed most of the charged particles in the brew. This means that the chemical interactions and possible changes are reduced to negligible levels, resulting in a stable beverage. Finally, it has an effect on making the brew look nicer in general. You will likely have seen cloudy beer, or even juice as an example. Juice that has been left on the bench in an open state will eventually turn bad. This is because things have gained access to it and turned into a pathogen. In beer, if it is bottled and still cloudy, this is because it has simply not been cleared, and as a result, it can often result in a less appealing appearance. Wine is seen as desirable if it is either clear and bright, in the case of white wine, or dark and smooth in the case of red wine. Beer is generally thought that it should be a darkish brownish colour to a light amber colour, but only a colour without any cloudiness or change in appearance. Thank you for watching this video. Please post any comments, questions or suggestions you may have below.